And thank you all for joining us. We love doing these updates for all of you, for our grassroots leaders and fundraising around the country. And I want to thank you all for your hard work and dedication in moving our mission forward. Those of you who have heard me, first of all, this is Jim Minow. For those of you who don't know me, Executive Director of the Cure JM Foundation. And for those of you who have heard me at the family conference or at chapter meetings, know that one of my foremost objectives is actually one that is now written into our mission, which is to find better treatments and a cure for juvenile myositis, with the emphasis, my personal emphasis at the moment, kind of being on better treatments. That really is, I think, our short-term objective, is to find better treatments so we can get our kids off of prednisone and steroids and other drugs with harmful side effects that you all know too well, onto replacement drugs, vamorolone being one that we think is a very promising prednisone replacement. We're not going to spend a lot of time, I don't think, talking about vamorolone tonight, but I did want you to all know that is high on our agenda for moving forward for a program, both a longer-term clinical trial for approval in JM and a shorter-term strategy to assure vamorolone's accessibility to patients with JM as soon as it is approved for another indication. We'll sort of put vamorolone in the parking lot for a future conversation, because there's a lot happening with our medical advisory board and with the clinical trial planning committee right now. So I think we'll have some good updates on that in the coming months leading up to the annual conference. But I do want to talk a little bit about an continued emerging emphasis on repurposing drugs that are already approved for other autoinflammatory indications, but which could work in juvenile myositis. We know anecdotally and through some small clinical trials that are either now being conducted or have been conducted in the past that these drugs do appear to work in some JM children, but we don't know which children that they're going to work in, and we really don't know a whole lot about the efficacy or exactly what the drugs are doing. And this is one of our goals in terms of really developing over the next 3, 4, 5 years and beyond, really a repertoire of potential drugs that we can then use and test in academic trials for proof of concept for their usefulness in JM. This whole approach, which has sort of been a couple of years in the making, in the thinking of our board, has really come to the forefront of where we want to take our research program. And I'm happy to introduce to you the man who's really going to drive this effort for us moving forward. He is our first Chief Scientific Officer, Dr. Andrew Heaton. He joined JM last fall, hit the ground running, is really focusing a lot of his time and attention on how, in fact, we bring together the various components, both academic researchers, commercial laboratories, and individuals and companies who have brought drugs through clinical trials and to the marketplace, and who know how this work for repurposing existing drugs is done. Dr. Heaton is a first-rate scientist and a chemist. He's developed 3 oncology drugs and taken them totally up to the clinical trial phase. And since joining CureJM, Dr. Heaton has really worked, as I mentioned, to align our research program with our research investments with a focus on drug repurposing as we move into 2019, 2020, and beyond. This looks at really making investments in better understanding genomics and proteomics of this disease, identifying how to upregulate and downregulate different genetic expression pathways, because not all drugs are going to work on all pathways, and so we think there's going to be several approaches and combination of drugs that are probably going to be necessary. As we know, this disease is different in every child. And finding approved drugs which show efficacy and predicting which kids will respond to which drugs. Again, not all of these responses in patients are going to be the same. So it's a monumental task. It's a huge task, but we're thrilled to have scientists of Dr. Heaton's background and track record join us to push this effort forward and to give you kind of an update on where we are and the path that we're beginning to move. I'm honored to introduce you all to Dr. Andrew Heaton. So Andrew, over to you.
Thanks, Jim. And thank you for your time on a Sunday night, everybody that's joined us. As you can pick from the accent, I'm not local. I'm from the other side of the planet, originally from Australia, having spent 20 years in the pharmaceutical area, taking drugs from in silico, so from a computer design perspective, all the way through to clinical trials. And so I've got a lot of expertise in drug discovery and drug development that I'm bringing to CureJM. And I guess my passion over the last probably 10 years has been rare childhood diseases, so particularly focused on some of the rare childhood pediatric cancers. So CureJM was a logical next step for me, sharing that passion with rare diseases and really trying to make an impact. So as Jim said, the object of tonight's talk is really just to go through, as I've been bringing myself up to speed with not only what the foundation's been doing, but who some of the key players are in JM research. And that's everybody from who's at the forefront in clinical practice and also who's at the forefront in really fundamental discovery. So I guess most of you would be aware, and I'll skip through some of these slides quite quickly, is the foundation actually polled the families as to, as Jim said, what is the most important thing that we should be doing as a foundation? And, they were a cure and better treatments always came up as top of the pops. One of them that came down a little bit lower, personalised medicine, I think that was from probably the way the questionnaire was constructed and from the way people probably understand what personalised medicine is. But hopefully by the end of this talk, I'll convince everybody that if we actually have new treatments, the new treatments are going to be reliant on making precision decision medicine decisions on drug treatments, which basically means there's going to be a lot of personalised medicine. As Jim said, JM is a spectrum of diseases. Every child presents slightly differently, so choosing the right treatments is always going to be an issue, and getting the diagnosis right. So as a foundation, the board basically put forward some key areas to look at how we could impact the disease, going from the right-hand side from clinical education, fellowships, ensuring that, standard of care. So those people that really are at the forefront in clinics are really providing, the best possible care to the kids. Then stepping back down, looking at drug discovery, drug repurposing, clinical trials, and really trying to expedite new treatments into the clinic. And on the left-hand side, which basically forms the grassroots of what we're trying to understand, is if you're going to accelerate scientific discovery, you really need to understand the disease in a great deal of detail. One of the things that I became aware of very quickly after joining the foundation was it's very standard in cancer therapy and certainly in cancer diagnosis that biopsies are routinely taken. Once a biopsy is taken, there's an awful lot of genetic and genomic information that's known on specific cancers and specific cancer types. So there's a lot of tailoring that actually goes on in cancer therapies. The same is not true with JDM. We don't have as much of genomic, proteomic and genetic history. So we really do need to bolster that. And while we're bolstering that, obviously we need to foster collaboration. It's a rare disease. There are not a great deal of people researching it, so we just need to make sure that those folks that are doing the research, we're not having people do the same studies over and over again. So getting people to collaborate across the globe is always a good thing to do. So I want to spend probably most of the time this evening just talking about how we can accelerate discovery and development for JDM, for better treatments. I think, as I said, it's not a single disease, but it's a range of diseases and a range of phenotypes. So there's a spectrum of kids that will appear. And while there's one broad diagnosis, there's going to be a range of phenotypes. I'm sure, as so many of you will be aware, some of the kids will present with more skin disease than others. Someone will have significant muscle weakness compared to others. So there's always going to be somewhat of a spectrum. So if we're going to accelerate discovery, really what we need to do and what we need to understand is for our JDM patients, we need to determine, first of all, what are the potential risk factors? So from a very fundamental genetics perspective, is there something or is there a range of genetic mutations that actually predispose kids to it? Now, we are doing quite a lot of work in this area. So some of you that would have been to our events would have seen the Children's Hospital of Philadelphia, or CHOP, taking a range of samples. They've got probably one of the world's best genetics labs in Philadelphia, and they're looking at mapping out genetics of patients and their families. The other thing we need to know is what cell signals, what pathways and what proteins are up and down regulated in the disease. So that's what we call looking at the genomics of the disease. So what genes actually get upregulated to actually create different phenotypes of the disease. And then looking at are there any proteins or cells that we can detect in the blood that will give us advanced warning of potential flares or some change in the disease. These are what we call biomarkers. So those are the grassroots and the real fundamentals that we need from building block perspective so that we can get better treatments. We have, as I said, we have gone some way in each of those areas. It's now about getting a little bit more data, or getting as much data as we can, and then melding that into a repurposing strategy. So hopefully this is going to work relatively smoothly. This is the Immune System 101, or Immune System for Dummies, and I just want to talk about it briefly and just highlight some of the issues that you've got when the immune system goes wrong. So just focusing on B cells and T cells, they're just two cell types, just two cell types associated with the immune system. One attacks invaders such as bacteria or viruses outside the cells. Those are the B cells. One attacks invaders inside the infected cells, T cells. And their role basically is they've got parts of the cell on the cell surface that recognize parts of the invader. Those parts of the invader they recognize are called antigens. So what they do is they effectively recognize the antigens. So just focusing on B cells, if we've got our bacteria, little skull and crossbones on him because he's bad, he shouldn't be there, there's going to be part of the bacteria that's recognized by the B cell. There's a couple of reasons why there's some question marks next to various arrows and things, and we're going to elaborate on those as I continue to talk. But what happens is once the antigen from bacteria has been recognized by a B cell. You then get a helper cell that comes along. The helper cell then basically, via a series of signal pathways, convinces the B cell to start rapidly dividing, forming plasma cells and what are called memory cells. The plasma cells then go on to produce a whole host of that same antibody that recognizes the antigen from whatever has invaded. Those antibodies will then basically link or neutralize the bacteria or the virus, and you will then get something called phagocytosis, where macrophages or killer cells will come up and basically destroy the bacteria. So that's what happens when the immune system works well. The question marks are basically indications whereby, you can have something that goes wrong with it. So the binding between the antigen and where the antibody is can go wrong. The cell signals to convince helper cells to come along can be dysregulated. Helper cells connecting with B cells can get dysregulated. So the take-home message here is that it's a finely balanced system, and small changes can throw it significantly off course. So one of the things that can throw it significantly off course is something called MHC, or major histocompatibility complex. This is basically, it's a protein that sits on the cell surface, and what it does is it presents the antigen to, in this case, T cells. So what it does, if you like, it's holding something out on the end of a fork for T cells to taste. So between the alpha-1 and alpha-2 units, that's where the antigen basically sits. And the way this is supposed to work, as I said, it's very finely balanced, whereby if you've got an autoantigen, so something that is of your body, you don't want that to interact with the T cells. Whereas when you've got an alloantigen or something from a bacteria or a virus, you do want that to activate the T cells, and you do want the T cells to go into a fight mode. So this MHC is critically involved in the immune system and presenting the antigens to the T cells. Linking this to JDM, one of the things that we do know is there's a specific genetic mutation that's carried along with a series of genes. The series of genes have got a couple of mutations, and they get carried along as what's called the haplotype 8.1. And in that, there's a specific genetic mutation with the major histocompatibility complex, whereby an arginine, as indicated here at position 74, gets mutated, the antigen doesn't bind correctly, and you get the immune system that basically throws everything out of whack, and it's no longer in that finely tuned balance. So what can go wrong in these autoimmune-based diseases is the antibodies that we generate from those B cells, instead of those attacking foreign antigens from bacteria or viruses, they're actually directed to a protein that's from the host. So they actually start developing cells and antibodies to specifically attack your own cells in your own body. That's obviously not a great thing to have, and that's the basis of autoimmune diseases. These autoantibodies, they tend to serve very much as a biomarker of the pathology of the disease. And the antibodies that you can actually detect, there are a whole host of different antibodies. And one of the things that's really coming to the fore, probably just in the last 4 or 5 years, is as we're getting techniques that are much more sensitive and can get a much better signal to noise, we're discovering more and more antibodies. The antibodies can be associated with myositis, which means that they could also be associated with other autoimmune diseases, or they could be specific to myositis. Just as an example, and I'm sure that some of you would be aware of these, different antibodies or different autoantibodies that we actually find give rise to different presentations and different prognosis. So having these antibodies recognised is really important to figuring out how the disease is going to progress. So anti-P155 is a specific antibody that's got a range of presentations associated with it. Anti-MJ, a different range of presentations. Anti-CAD140, different, and anti-HMG-CoA. So these are all antibodies that are associated with the disease. So these autoantibodies are dysregulated. They're appearing where they shouldn't be appearing. Their regulation is somewhat out of control. So those little question marks that I indicated in the little immune disease system 101 slide, those question marks, they're all potential control points in the immune system that could go wrong. So really, just in the simplest of terms, what we're trying to do when we're trying to develop any new drug is we're trying to normalize anything. So be it an antibody, be it a specific protein, be it a specific pathway, we're just trying to restore things to normal. So just in the simplest specific example, we could have a pathway that's upregulated from normal. That could be a pathway, it could be a cell, it could be an antibody. The object, once we've actually diagnosed or once we've actually determined that a pathway it has been dysregulated, is then to find a drug that re-regulates that specific pathway or cell type. So it's literally, it's, you know, you add one to the other, and what should magically happen is you should magically re-regulate the pathways. That's a very simplistic view, as you would appreciate. But with JDM, because there are so many different phenotypes, we're going to have so many different pathways and cell types that are dysregulated. So basically here what I've indicated is that even just considering seven different pathways that are dysregulated, some of them are upregulated, some of them are downregulated. The treatment for a number of dysregulated pathways, more often than not, is going to take not just one drug, but you're going to need to take two drugs that will actually re-regulate each of those pathways. So adding the two drugs, we end up, hopefully at the end of the day, with completely regulated pathways. So basically the take-home message here from these first series of slides is that, you know, the fundamental understanding of the disease process and what's actually happening at a molecular level, at a pathway level, and at a cellular level is really critical to figuring out, you know, how much pathways are dysregulated. Once we can figure out which pathways are dysregulated, we can then make precision decision on which treatments we should actually use. So we should actually be looking at which drugs are more likely to work best with individual patients. So really what I'm interested in doing, and as Jim said, you know, really what my passion and my vision for the next few years, and certainly building on the excellent work that's already started, is really looking at the genomic data that we've got. As everybody's aware, JDM is a rare disease, so generation of genomic data requires access to appropriate samples. The samples that we need access to need to be really very well clinically annotated. So the samples, you know, we need to know, you know, is it a newly diagnosed patient? What was the state of their disease at diagnosis? Have they had any previous treatments? If they were on treatments, what treatments were they on? So we need to acquire samples that have got as much information as possible. Then the other critical thing that we need to do with patients is, rather than just having just one sample from each patient, is to actually have samples whereby we follow them longitudinally. So we can follow kids longitudinally. What that does is that gives us great insight to, you know, the following pathways and cell types were dysregulated at the start of the disease. They went on to steroids. These pathways got re-regulated. They were on a variety of drugs for the next few months, and this is what happened to it. So actually having those longitudinal samples is going to be critical to help our understanding of the pathways and the dysregulated pathways. One of the things you're going to be hearing of more over the next few months is one of the most inspired decisions the board made in the last year was to fund what's called the CARA biorepository. So the Childhood Arthritis Organisation have basically been sampling kids as they've been presenting in hospitals that are associated with the CARA organisation. The idea behind this repository is very much to just look at patients that are newly diagnosed, attempting to get those samples that are clinically annotated. So how did the patient present? And then following those patients with very detailed information over the course of the disease and hopefully over the course of the resolution of the disease. So we can learn more about each individual and how each individual responded to treatment. But with those blood samples and other samples we get from each kid, we can actually look at how do we actually get the best drug cocktail for each individual. The other thing that, as I said, the autoantibodies that we look at, because they're associated with different pathogenesis of the disease, it's really important that we actually get those antibodies analysed. And I'm sure, as a lot of you are aware, that the routine analysis of some of these antibodies is actually lagging behind their potential utility. And again, one of the inspired things the Foundation has done in recent history has been to fund Bath University in the UK, where they're looking at adapting some methods for antibody analysis associated with JM, with the ultimate end goal of having specific analytical techniques that they can roll out globally into a range of labs just to make it much more routine to actually analyse for specific antibodies. That's obviously going to aid in treatment decisions as well. So not only looking at what antibodies are specific within the disease, the other thing we're actively looking at is building up this genomic signature and getting what's called a bioprofile from each kid. So there's a specific genomic technique where basically people print out little bits of DNA onto a slide, basically print out the whole genome. You can extract RNA from the blood from an individual. You basically hybridize, label the blood, put the blood onto a slide. You then read it with a laser. And effectively what you get out is what's called a heat map. So you get what the status of each particular gene is in an individual. So how much is that gene being turned into protein? And obviously, if we look at a range of not only healthy kids, but sick kids, what we'll then be able to do is we'll be able to form what's called a cluster analysis. So for each phenotype of the disease, our goal is to actually have a specific heat map whereby we know exactly which pathways are upregulated, which pathways are downregulated. Once we know which pathways are upregulated and downregulated, there's then a number of companies globally that can actually figure out from the drugs that are already on the market which pathways they regulate and re-regulate. So that will aid us in making decisions as to which drugs are most likely to work, not just within JDM, but critically for a specific individual. So if we do cluster analysis on the different phenotypes of JDM, hopefully we'll be able to get to the state of having a blood test that's going to aid in us making decisions as to defining what treatments we should actually be using. The key here is obviously getting the best possible genomic data that we can. That's the longitudinal samples, looking at the samples with the best possible techniques, and we believe we have partnered up with a company that can actually do that effectively. So the idea is, and this is my my last slide. So I've left the most complex for last, but just to highlight what we're doing and what others are doing as far as JDM and JDM pathways are concerned is a lot is already known as to, B cells and T cells and what's integrated and what's dysregulated in JDM, what antibodies drive certain pathways. And because we know an awful lot about some of the pathways that are dysregulated, we've already started the repurposing program. So things like abatacept and rituximab, I'm sure many of you have heard about the rituximab studies. We're looking in much more detail in the rituximab and looking at exactly why did some patients respond really well to rituximab and some patients not respond quite so well to rituximab. So looking at which pathways are dysregulated will help us make better decisions as to when we should be using rituximab. Looking at the major histocompatibility complex, we're actively looking at a program with NCATS in DC, part of the NIH, where we're looking at repurposed drugs and which drugs can help us with that major histocompatibility complex potential issue. So the gap we're really looking to fill over the next 2 to 3 years is, besides the pathways that we're already looking at, where are the gaps and what other pathways should we potentially look at? So we're going to dig deeper from a genomics perspective, dig deeper from an autoantibody perspective, so we can really practice that precision decision treatment option. And I think the idea behind digging deeper into what signals are dysregulated, if we have the most sensitive way of looking at the genomic signals, that's hopefully going to uncover some pathways that are outside what we currently think. So outside the square, outside the box, depending on which country you're from. We're going to think outside the square or think outside the box to look at pathways that really haven't been thought of yet, that have got some potential validity as to a repurposing program. And this is all going to be based on, as I said, we need a fantastic repository with those longitudinal samples. We need high quality antibody analysis. We need high quality genomic analysis, and critically we need obviously extensive information on the clinical annotation of the samples from the patients, so we know how patients have been responding to various drugs and drug treatments. So as I said, we already have a very detailed program looking at rituximab, and we're going to try and determine exactly why that is and why that isn't working. And now, just because Jim likes quotes, I'm going to finish from a famous UK Prime Minister. Winston Churchill said, now this is not the end. It is not even the beginning of the end, but it is perhaps the end of the beginning. So I think, as far as JDM is concerned, as I said, we've got a lot of really good solid background information that we're building on. So we've started the repurposing strategy. It's now about making better decisions with repurposing, looking deeper, getting a broader sample set, and really looking outside the square to try and get any better drugs. And at that stage, I'm happy to address any questions anybody has.
Thank you, Andrew. That was a terrific summary of where we are and where we're going, and we're very appreciative of course, your leadership and the leadership of the Medical Advisory Board. I know many of you know our Medical Advisory Board, at least some of the members, which who are working with Andrew to push this agenda forward. And as a team, I have a great amount of confidence that this group is going to move us, past the walking stage and the early stages of drug discovery and drug repurposing to be at a full pace and a full run here in a very short period of time.